Myself, Mr. Mate H.T., Department of Botany, RBNB College, Sri Rampur. So today we are going to study the T.Y.B.Sc Botany, paper first, BO 331, that is a cryptogamic botany. And from this topic number 3, that is the life cycle of the Chara. And from the life cycle of Chara, today in this lecture we are going to study the reproduction. The Chara reproduces by two different types of the methods, that is vegetative and sexual. And here in this Chara, asexual reproduction is completely absent. So the Chara reproduces only by vegetative and sexual reproduction. Now from this, number 1, that is the vegetative reproduction. Now vegetative reproduction is again of different types. From this, number 1, that is the amylum stars. Now these are the star-shaped structures. They are called as the amylum stars because they are star-shaped in outline, in structure, and they are full with the food material, that is the amylum starch. So they are called as the amylum star, and they are developed on the nodal region of the lower nodal region of the main axis. And when they get detached from the parent plant, they have capacity to develop into a new thallus of the Chara if the conditions are favorable. Second, that is bulbils. These are the bulbils. They are also the vegetative reproductive structures. They are also full with the food material. They are circular or tube-like in outline. They are developed on the rhizoid and also on the lower nodal region. And when they get detached from the parent plant, they have capacity to develop into a new thallus of the Chara. Now third type, that is the amorphous bulbils. They have no any proper shape. They are irregular in shape. They are just like the bunch. They are developed on the lower nodal region of the thallus. And when they also get detached from the parent plant, they also develop into a new thallus of the Chara. And fourth, that is the secondary protonema. Now these are the secondary protonema. They are filamentous. They are developed on the rhizoids of the Chara plant. Now on the rhizoid, primary protonema are get developed. From primary protonema, secondary protonema are get developed, and from the secondary protonema, the main thallus of the Chara plant is get developed. So these are the 4 different types of the vegetative reproduction which are found in different species of the Chara, that is amylum stars, bulbils, amorphous bulbils, and secondary protonema. Next point, that is the sexual reproduction. As the Odonia are dominant in the life cycle of the Chara, the type of sexual reproduction in a Chara is oogamous. So the type of sexual reproduction is oogamous found in the Chara. Now the plants are monoecious or dioecious. In Chara, there are the 2 different types of the thallus are found on the basis of presence or absence of the reproductive structures. For example, monoecious. That means here both male and female reproductive structures are developed on one and on the same filament. Such type of species of Chara are called as the monoecious species. And when the male and female reproductive structures are developed on 2 different plants, then such type of species are called as the dioecious species of the Chara. Now here the male reproductive structure is called as the antheridium or globule, while female reproductive structure is called as the Odonia or nucleus. So this structure or position of this globule and nucleus is specific and it is found in the Chara. So it is only found in the Chara. Now the position of reproductive structures. Now this is about the position of the reproductive structures. Now this is found in the monoecious type of the thalli. That means both of the reproductive structure, that is Antheridia and Odonia, are developed on one and the same plant. So this is the primary lateral branches and these are the secondary lateral branches. And in the axil of this secondary lateral branches and on the nodal region of a primary lateral, they are developed the reproductive structure. And here in Chara there is a specific characteristic feature, that is female reproductive structure is toward the upper side while male reproductive structure is toward the lower side, and this is found in Chara only. Now next one, that is the structure of the globules or structure of the Antheridia. So this is the structure of the Chara. It is developed on the nodal region of the primary lateral and in between the axil of the secondary lateral. They are developed on a rod-like cell. That cell is called as the pedicel cell, and it reaches up to the center of the antheridium or the globule. Now this antheridium or globule, it is spherical in outline. It is yellow or green, and it is about 1 mm in diameter. And outer side it has a cells. These cells are called as the shield cells. These shield cells, 4 are from front side and 4 are from back side. That means that they are 8 in number. So shield cells are 8 in number. And from in each shield cell there develops again a rod-like cell. They are directed toward the inner side and they are called as the manubria. So manubria are also 8 in number. Now each manubria, that means all the 8 manubria, cuts off a small cell at the center. And these cells are called as the primary capitula. They are called as the primary capitula. So they are also 8 in number. Now this primary capitula again cuts off a cell, single cell. That cells are called as the secondary capitula. And in some species of the Chara also tertiary capitula are also get developed. But finally from secondary or tertiary capitula there are developed small filaments. And these filaments are called as the antheridial filaments. They are branched. 2 to 3 filaments are developed from secondary or tertiary capitula. They are branched and each cell is get metamorphosed into a structure. That structure is called as the antherozoid. So this is the antherozoid. Now in each filament there are about 250 cells are there, and from each cell there develops the antherozoid. So this is the structure of the antherozoid. This is the coil. It has 2 anteriorly inserted flagella. So these are the flagella. So this is about the structure of the antheridium. Now when antheridia becomes mature, when antherozoid becomes mature, they gelatinizes the wall. That means they gelatinizes the wall of the antheridial filament. And a small slit is get formed in between the shield cells, and all of these antherozoids comes out in the water and freely swims in the water with the help of the anteriorly inserted flagella. So this is about the liberation of the antherozoids. Now the structure of the nucleus, that is the Odonia. So this is the structure of the nucleus. They are also developed on the nodal region of a primary lateral and in the axial of the secondary lateral. They also develop a small cell which is called as the pedicel cell. This pedicel cell is get attached to the nodal region of the primary lateral, and above this there is another cell which is called as the central cell, and above the central cell there is the stalk, and above the stalk there is very large oval shape which is called as the egg proper or the ovum. It is called as the egg or ovum. Now this egg or ovum, it is get spirally encircled by another kind of a cell, and these cells are called as the tube cells, and they are 5 in number. Now these tube cells at apex cuts off a small single cell, and it is in the form of a corona. So in the corona there are again 5 cells. That means the corona is formed from the 5 cell. Now just below the corona there is a colorless spot which is called as the receptive spot. Now it is a oblong or oval in shape, and its length is about 7 mm. That means its length is 7 mm, and it is a green or yellow in color. Now this is about the structure of the nucleus, that is the Odonia. Now next point, that is the fertilization. When antherozoid becomes mature, they freely swims in the water, and by chance they comes in a contact with this receptive spot. Very large number of the anthrozoids comes in contact with the oogonia or the egg, but finally only one enters in the egg and union of the male and female nucleus takes place, and there is a formation of the zygote. Now zygote has a single diploid nucleus. After fertilization, zygote has a single diploid nucleus. Now when this zygote forms a thick wall around itself, it is called as the oogonia. So it is called as the oospore. Now when oospore is get formed, it is get settled at the bottom of the water reservoir, and after taking the rest, it starts to germinate. Now the germination of the oospore. The oospore is the black or brown in color. It is get modified from the egg. Now this is about the germination of the oospore. Now initially the oospore has a single diploid nucleus. Now this nucleus goes toward the upper side, that means towards here. There it divides meiotically into the 4 haploid nuclei. That means these are the 4 haploid nuclei. Now they are get divided into the 2 groups. 1 nuclei goes toward the upper side and 3 nuclei comes toward the central, and in between these 2 a wall formation takes place, and there is a formation of a 2 unequal cell. The upper cell is small while lower cell is the large. Now the 3 nuclei which are in the lower cell, they get degenerate, and only 1 nuclei which is here, that means the upper cell, it becomes functional. Now this cell also divides vertically into the 2 cells. From this, 1 cell is called as the protonematal initial cell, and another cell which is somewhat toward the lower side is called as the rhizoidal initial cell. Now this rhizoidal initial cell develops into the primary rhizoid, while the upper cell, that is the protonematal initial cell, develops into a primary protonema, and it has only the 2 nodal regions. It has only the 2 nodes. Now from this, that means these are the 2 nodes, they are developed in the primary protonema. Now from lower node of this primary protonema, they develop the permanent, that is the secondary rhizoids. And from the upper nodal region, they develop the main axis of the next Chara plant. In this way, a new Chara plant is getting developed from this nodal region of the primary protonemata. Thank you.